Let's get into epithelium. Epithelial tissue. Epithelium is going to cover exposed surfaces and internal cavities and passageways. Okay? Or epithelial tissue is going to be making up glands. Okay? There's two types of glands. Two general types. We can have a gland that has a duct that leads to an open cavity. Its secretion is made in the gland but goes through the duct and it enters this open cavity. This could be your salivary gland, which has a duct that leads to your oral cavity. When it has a duct like this is called an exocrine gland. We also have another type of gland that does not have a duct. Instead, this gland rides alongside a blood vessel, and its secretion goes from here and goes directly into the bloodstream. This secretion is always going to be called a hormone, and this type of gland is called an endocrine gland. Things over here is anything but a hormone. Could be enzymes, could be water, could be mucus, but not hormones. Hormones go directly into the bloodstream. Okay? So, epithelial tissue could be epithelial or it could be glands. If it's glands, it could be exocrine glands or endocrine glands. Right? Trying to keep this organized. It's not bad. It's a monster, but it's not bad. Just keep it organized. Glandular epithelium. We could describe glands in different ways. We could say that it's made up of, MUO is made up of one cell, or it could be made up of many cells. It could be simple, meaning the duct has no branches, or we could say it's complex or compound, and the duct has many branches. It could be that the gland and duct are the same diameter, or it's going to be the gland itself will be a cinder or alveolar, which means it's more like globular, more like a bowl. Let me give you an example of pictures. Here's a simple tubular. The gland part is here, the duct is up here. See how it looks like it's the same diameter. It's simple. It's got no branch really. This one has a few branches on it, so this is compound. Here's simple, meaning like it's not branched, but it's a cinder, so it has like a ball over here. Or you could go into a compound cinder and it's kind of a mixture. So it's not bad. They have names though. Other glands. This is also on your dermatology lecture too, okay? One that you'll do on your own. We have merocrine glands. All right, all of these are exocrine glands. We have a merocrine gland, which is fluid that's released through the cell membrane through exocytosis. Good example of this is salivary glands and certain sweat glands. What's happening here is, if you look up here, it's animated. And what's happening is, this blue, that's the cell, and that blue thing is the nucleus. What you see inside there is a vesicle with proteins in it, whatever it's going to release. So this glandular cell is going to release that red stuff. How? Well, the vesicle itself is going to become part of the cell membrane and immediately will release all the contents from it, exocytosis. An apocrine gland is where a portion of the cell membrane gets pinched off with the secretion, and later on it gets released. It does this kind of thing with the, if you watch the animation on here. A good example of this is a mammillary gland and certain other sweat glands. See how the cell membrane becomes a part of the secretion itself, so it doesn't immediately get released. Then we have a holocrine gland, and this is where the whole gland explodes. Okay? Sebaceous or oil glands are a good example of this. And the whole thing will explode, including the nucleus. Okay? Now, let me ask you this. Of the three here, which one, which two, all three, or none of them, which one will have DNA parts in its secretion? Which one is going to release it? The holocrine one. Look at it. It explodes. So you're going to have pieces of DNA here. Here, the nucleus stays the same. It doesn't get ruined. So how do you use that? Well, in forensics, oil glands, that's where it's going to have your DNA on it. So if you're thinking about murdering someone, please put gloves on. All right? Not that I'm encouraging that. Oh, let me tape myself. No, I'm not encouraging. But how do you explain over here? Well, geez, things over here like the mammillary gland. Yeah, but the thing is, when they do that, like in the salivary glands, well, yeah, when someone has saliva on something, you get DNA there, but that's because you're getting some of the cheek cells in the saliva. So, but if you get, I don't know if you could say distilled saliva, if you just get the saliva coming out of the gland, there's no DNA in there. But because it gets mixed up with the cheek cells, yeah. So really, you can't get away with murder. Okay? But anyway, that's what I want to say about that, all right? All right. And this is just showing the same thing, but in picture form instead of the animation. All right, now, functions of epithelium. They provide physical protection. They control permeability. If they're the lining of your stomach and the lining of your intestines, it's going to control what can get absorbed into these areas. They're going to provide sensation and produce specialized secretions, as you can see, because they're also glandular. Now, basic features of epithelium. Let me actually start doing some more of this therapy for you, art therapy. And I want you to see how this is all coming together with the 4 different tissue types. Where does epithelium come in contact or play a role with muscular, and where is it actually seen over here? So let me show you what's going on. Let's get a tubular structure. Any tubular structure. Stomach, doesn't matter. Blood vessel. So we're going to look at the end of this over here, and the end is going to have this kind of setup. These dots that I'm making are the nucleus of each one of these cells. I know you guys can't see it, so let's make it bigger. Okay? See how I'm drawing it so far? Alright, now, you have this tubular area, this space over here. This space has a special name. It's called a lumen. The lumen of a fire hose is where the water goes through. The lumen of blood vessels where the blood goes through. Okay? It's that cavity. It has the name lumen. Okay? Then you're going to have this epithelial tissue that's lining that lumen. So that's where the epithelium is. Okay? Around there, you're going to have connective tissue. Okay? Around that, you're going to have muscle. Scattered all throughout that area is your nervous tissue. And the muscle and the connective tissue. Overall, that's where we're looking at. Okay? Now, let's go a step further. There's your epithelial tissue. Connective tissue over here. Muscle tissue here. In between the epithelial tissue and the connective tissue, that's where we're going to have our basement membrane. This orange line that I'm drawing here is the basement membrane. Epithelial tissue, at least the lowest layer of epithelial tissue, is going to be attached to this basement membrane. This is your basement membrane. The epithelial tissue, the side that is facing the lumen, or the outside world, the uppermost layer, that's called the apical end. Everything I'm saying here is going to be on the next few slides, words-wise. This will be the apical end. This will be the basal end. Okay? The apical end may be flat like that, or there might be certain things there. The apical end may have cilia. And the cilia would move, right? Okay, some of them wouldn't, but when we're talking about here, in this epithelial thing, they would move. Okay? Like in the trachea. They would, the windpipe is going to move the mucus up so it can cause a coughing reflex. Some of them may have something called microvilli. Look like cilia, but they're not. They're there for a totally different reason. These don't move. They just stay there. What they do is increase surface area. At this point here, if I was going to have absorption going on, the only place I could absorb things is here and here and here. But if you have these microvilli, microvilli, you can have absorption happen here, but now you can have absorption happening here and here. You're increasing the surface area so that you have more absorption occurring. Does that make sense? Okay? Surface area is this. If I take this structure here, the only place that something could actually touch is the outside surface. Now, without changing the volume of this creature that I have here, I can increase the surface area though. Watch. I created 2 new surface areas that things could actually absorb to. 2 new surface areas. If I take a block of ice, it has a surface area, and I put it on a stove, it will actually melt. But the only place it's going to melt from is the place that actually is on the outside. It can't go from the inside, it can only be on the outside. But if I take that same block of ice and chop it up into ice chips, there's a lot more surface area that we have here. If we put that on the stove, that's going to melt faster, is it not? Because there's more surface area. Okay? Does that make sense? What surface area is? I know people always hear that, but you've got to see it because we're going to be dealing with surface area for the rest of A&P 1 and 2. Okay. So it doesn't have both. It's either going to have cilia, microvilli, or nothing at all. All right? I just put this all here. I created a new creature. It doesn't exist, but just put it all on here. Okay. So, like I said, the apical surface is facing the lumen. It could have microvilli, cilia, or nothing. Okay? The base is going to be the lower part, and it's going to attach itself to the basement membrane by way of those hemidesmosomes. And that's going to attach to the connective tissue. Epithelial tissue is avascular. There is no blood vessels there. So how does it get its nutrients? Well, the blood vessels are down here. The oxygen and nutrients are very concentrated up here. There's nothing up here. Diffusion. It's going to go from a high concentration to a low concentration. That's how it gets its nutrients. Okay? By simple diffusion. They have a high cell turnover, meaning that from 1 cell turning into 2 cells, that rate is very fast. We're talking about seconds to minutes to hours. That's very fast. 1 cell turning into 2 cells. Your skin is not the same skin you were 4 years ago or 5 years ago. It's new skin that developed. The significance of knowing that has a high cell turnover, if I want to make 1 copy of your test upstairs on the Xerox machine, what's the chances that something's going to go wrong with that 1 copy in the machine? High chance or low chance? What's that? High chance. 1 copy, just making 1 copy. Low chance, right? What about if I need to make 1,000 copies? What's the chances that the paper's going to get jammed up in there, one's going to be blank, it'll shoot like 2 papers out there, it'll crinkle? What's the chances if I do 1,000 copies? Much higher. So when you have a high cell turnover, going through mitosis that rapid, there's a greater chance that it could get mutated. Does that make sense? And mutation should ring a bell to you, and the next word that you should be thinking about is cancer. We are seeing more cancers than ever with epithelial tissue. How much? 85% of your cancers that are out there are epithelial in origin because it has such a high turnover. Which ones have a low turnover, cell turnover? Nervous tissue, muscle tissue. I mean, 1 neuron, nerve cell, turning into 2 nerve cells take about 70 years, if that. So 1 nerve tissue is not going to really turn cancerous. You don't really hear people having brain cancers. But I do. No, not nerve tissue, but I hear brain cancers. Yeah, because they're going towards the public. The TV is doing that, brain cancer. It's the truth, it is, but ask what type of brain cancer. Well, it's a pituitary gland tumor. What did you say? Gland. Then what is a gland a part of? Epithelial. Do you see what I'm saying? The neuron itself is not going to become cancerous. It's extremely rare. And when was the last time you heard someone got a muscle cancer? Okay? It's a little bit more rapid than nervous tissue. All right? I have seen, let's say the uterus becoming cancerous. All right? Not the lining of the uterus, endometrial cancer, the lining of the uterus, that's epithelium. But the muscle itself, rare. But it can happen. Okay? Nerve tissue does not become cancerous. It's highly unlikely. Let me step back. There is one that children get where the nerve cell actually becomes cancerous. It's called neuroblastoma. But that's because in a child, their nerve cells are still developing. So they're still going through mitosis at that point. All right? And I'm talking about like a toddler. I'm talking about very young. Okay? But you don't see cancer happening with nervous tissue and muscle tissue. Connective tissue a little bit more, about 10%. But 85% of your cancers are epithelial origin, okay? So high cell turnover, regenerate, reproduce quickly. Think of it. Look at all the cancers that you're hearing out there. Melanoma, skin cancer. Basal breast cancer, that's glandular. Intraductal cancer, that's glandular. Lung cancer, the lining of the lungs. Colon cancer, the lining of the colon. All right? So that's why I'm trying to show you, you need to know where these, like I put it together for you in that chart, you need to know the locations of some of this stuff because it's going to help you in your future to understand why you're getting all these cancers. And if you go into research later on, that's why it's happening, unless you go into cancer research. Okay? So 85% of your cancer occurs from epithelium. If it ends with the word carcinoma, all right, basal cell carcinoma, intraductal carcinoma, all right, which is breast cancer. If it ends with carcinoma, it's an epithelial cancer, okay? Questions on that? I spent some time on it. Usually when I spend a lot of time on something, that's my way of saying hint, hint, it's going to appear someplace on the test, okay? All right, so nomenclature epithelium. If the cells are thin, we say it's squamous, thin and flat. If they're in boxes, they're called cuboidal. If they're in columns, we call them columnar, rectangles. Okay? If there's one layer, we say it's a simple epithelial tissue. If there's many layers there, then we say it's stratified, many layers. Okay? Let me show you a few things here. Okay? One layer, very thin and flat. Remember, the dots I'm trying to show is nuclei, okay? Very thin. This is one layer, so we say it's simple. Because the cells are flat, we say it's simple squamous epithelium. Okay? They're cubes, meaning the nucleus is dead on right in the center. It's the same distance as it is widthwise or lengthwise. That's cuboidal. And if it's one layer, we say it's simple cuboidal. In columns, the nucleus is usually pushed down to where the basement membrane is, in that area. It's pushed down there. It's not in the middle. This is columnar. And if it's one layer, we say it's simple columnar. Okay? See, it's not bad. All right? Now, sometimes we're going to have, if this is going to be your basement membrane over here, we have many cells that are going to be all around here, different sizes and shapes. Difficult to say what it is. It could be squamous, it could be cuboidal, it could be columnar. But what's going to happen, how you're going to name it, we know that there's a lot of layers there, so we know it's stratified. But how can you tell if it's stratified columnar, cuboidal, or squamous? You go by the most apical cells. If they're thin up there, then this is going to be stratified squamous. If they're cuboidal up there, it's stratified cuboidal. And columnar, it's stratified columnar. So you go by the uppermost layer. Okay? This is basically a chart form. The different types over here, the description of how they would look, and the locations. Okay? There's two that I don't want you to really, I want you to know they exist, but I'm not going to emphasize them. Stratified cuboidal and stratified columnar. They're very rare. Stratified cuboidal is found like in the ovary. Stratified columnar in the urethra of the penis. And they're very difficult to actually see. Know that they exist, but I won't test you on them. Okay? So far, so good? I'm going slow for you. Okay. Now, these came from your lab books. These are epithelial tissues with some images. This is simple squamous epithelium. You need to know, on all these kinds of tissues, you need to be able to identify them. You need to know, give me a location that you would find these in, an organ that you would find these in. And you need to know the function of them. And if there's any specialized cells, which we'll go through as we go along here. This is simple squamous epithelium. Let's take, for instance, this, what I just drew here. If this is an air sac of a lung, okay, and it's going to ride alongside a capillary, which is a type of blood vessel. Here's an air sac. This is a blood vessel. I'm going to draw green, but this is a red blood cell that's going to be inside this blood vessel. You getting the picture? Okay? What happens here is this. Oxygen is going to come in here and need to get onto the red blood cell. Because like I said, hemoglobin is going to carry that oxygen for you in the red blood cell. So this oxygen is able to go right through a very thin layer. You would expect it to be thin. You don't want it to fight through a thick layer like a cuboidal or columnar. So it passes right through a very thin layer, and there's another layer here on the blood vessel that's also thin. And it will pass right through there and go right on there. So what's going to happen is, in the lungs, you will have simple squamous. In blood vessels, you will have simple squamous. Why? Because you need gas exchange to occur there. You can't have it very thick. This is the thinnest possible. Again, physiology reflects anatomy. Function reflects structure. It makes sense why it's doing this. And it's simple diffusion. We have carbon dioxide. That's over here. High of dosage in here, low dosage here. it's able to pass through here and pass through here to get out. So the function of simple squamous epithelium is simple diffusion. Or you could say gas exchange. Or when you get into the kidneys, filtration. You want, let's say, your coffee to be going through that filter, which is really thin. You don't want to make it go through styrofoam. It'll never get through there. Let me just introduce you to, I won't test you on this, but I want you to really understand the concept. In someone who's got pneumonia, what happens here is that the alveolar cells, these cells here, these thin cells over here, get inflamed. It gets infected with a bacteria that causes pneumonia. That's what pneumonia is, infection in the lungs. And what happens here is that you learn about inflammation, but these cells swell during inflammation. You ever have, you stub your toe and it swells, or someone who's got arthritis, their joints swell? That's inflammation. It adds water. Water starts building up in here, and the water will actually go into this area. The water is going to bring in a lot of our phagocytic cells, white blood cells, to fight that bacteria. That's what's happening. But look what's happening here. These become thicker, and this also fills up with fluid. Now, this oxygen needs to still get over here. So it comes here and like, oh my God, I gotta pass through all of this water. And then go through these thick areas here to get on here. It's very difficult. This is why people are dying with pneumonia, because you can't, everything gets inflamed there, gets thicker. So this is really showing you that you need thin tissue, simple squamous epithelium, for good oxygenation, good gas exchange to occur. Okay, is that clear how to use this? Okay. So you can see here, and this is at 400x. Very thin. That's like one cell right there. There's a cell over here. This is an alveolar sac. Very thin over here. Okay? So I could point this out to you and say, what is this tissue? You would say simple squamous epithelium. Where would you find this tissue? Give me an organ you would find this in. You could say lung. You could say blood vessel. You could say kidney. Just pick one. All I would ask for is one. Pick one. Because if you put two things there and say, well, it's in your skin and it's in your kidney, it's in your brain, don't run if you're not being chased. All right? Just put down one and be confident about it. Because if you put down more than one thing there and one thing's wrong, the whole thing's wrong. Don't make it into a multiple choice for me. Okay? Question?
So, the line, is it saying that the lines are the cells? Not...
This is the line itself is the cell.
Okay, not...
This is an air sac.
Okay.
This is where oxygen is. This is what I was trying to draw here.
Okay.
But this here, this line, each one, if you look at really close, but you can see the nucleus right there, nucleus over there. They're really thin, and that's emphasizing how thin it is. Okay? Questions on simple squamous? Now, I want to do that fairly slow with you so you have an idea of how to kind of put it in your head, because we have a lot more to do. But it's not bad. All right, so it's just showing you how thin it is, and you can see the lungs over here. All right, this is simple cuboidal epithelium. We're doing a cross-section of the tubes here. This is in the kidney. So we're doing a cross-section here. All right, there's a tube, there's the lumen, there's the lumen, there's the lumen. There's a lot of tubes here. Now sometimes you're seeing a nucleus there in the cell, but there's nothing here. That's because of the way it was cut. If I was going to take an egg and the yolk is over here, if I slice it here, it's going to look like that. If I slice it here, it's going to look like that. if I slice it here, it's going to look like this. Does that make sense? So it depends on where it's getting sliced. You got to think three-dimensionally. Maybe it's an oblique cut here and it's not getting the nucleus at this point here. All right? Not a big thing, but I don't want you to get confused, like, why am I seeing dots but not all around? It's because of the way it's cut. Okay? So this is simple cuboidal epithelium. You'll find it in the kidney. It's going to be there for secretion and absorption. Okay? And it kind of looks like that. Now, here's simple columnar epithelium. Simple columnar epithelium is going to look like this. The nucleus down here, I have it right there. Sometimes we're going to have cilia up here. Sometimes you may have microvilli. If microvilli are there, then it's going to be in the digestive tract, specifically from the stomach all the way down. Not the esophagus, not the oral cavity. So you can't put down, if I ask you where it is, you can't say digestive system, because that's a, you're talking about the salivary glands, you know what I'm saying? Are you talking about the esophagus? You got to be specific. Stomach, large intestines, small intestines, that's fine. The gallbladder too, for that matter. These would have microvilli because that's where you're going to have absorption taking place. They may have cilia on it, and that's where you're going to find simple columnar epithelium located in the fallopian tubes. Okay? And the fallopian tubes will have the cilia there to try and push that egg towards the uterus, where the sperm is. Like I said to you, come on, it's okay. He's a good guy. You gotta go over there. He'll make fun. It's a little. They're like the little cupids. You get the two together. Okay. Now, also you may find something called goblet cells. Goblet cells kind of are shaped like this. If you know what a wine goblet looks like, it kind of looks like this. Yeah? So it kind of looks like this. These are filled with mucus, and it spews out mucus out there. Mucus kind of repels stains. So because there's mucus inside, they don't really stain well. They become little white balls. You can't stain them very well. They need special stains. So to me, they start looking like little Casper the Friendly Ghost things just floating around there next to the simple columnar epithelium. But that's where you're going to find goblet cells. You'll find it in the simple columnar epithelium. Okay? And you can see here's columnar cells. The nucleus kind of pushed down. Microvilli is up there. That's what it'll look like, microvilli. You really have to get up close to really see these little finger-like projections. But there's a wonderful goblet cell that's going to spew out mucus. Okay? Schematic kind of drawing of simple columnar epithelium. Stratified squamous epithelium. A lot of layers. You can either have it keratinized or not keratinized. This will make more sense to you when you do the dermatology. But very briefly, keratinized is that it's going to be tough, water resistant. So you're not going to lose water over here. Okay? So it's going to be on your skin. Then we have non-keratinized, a tough, and it can dry out. These are areas that are in your oral cavity, it's in your esophagus. So we have a lot of mucus that's going to be secreted out over here to keep the area moist. Keratin is a protein, and like I said, you'll learn more about it when you do your dermatology. Now, a lot of layers. See how they're different sizes and shapes over here. But when you look at the uppermost layer, they're thin. So this is stratified squamous. A lot of layers there. So if you have a lot of layers, think of where you would expect it to be. You would expect it to be on your skin. You would expect it to be in your esophagus. Why? Because it's there for protection. When you touch something hot, you don't want to have simple squamous epithelium on your skin. It'll burn a hole right through there, go right into your connective tissue, and burn a hole right into your muscle. You need some protection there. When you drink something hot, you would expect your esophagus, the tube that goes from your mouth to your stomach, to have a lot of layers there. So when you swallow, it won't burn a hole right through there into your muscles that are surrounding your esophagus. So in one word, what the purpose of stratified squamous epithelium is protection. You would expect it to be there. You wouldn't expect it to be in the lungs. All right? Again, physiology reflects anatomy. Think about it. Instead of just memorizing, think about it. Okay? And this is where it's showing on here. Now, there's two more I have to do, and I'll give you guys a little break. Okay? Those are the easy ones. Actually, this is easy too. We have something where we have, here's the basement membrane. Then we have more cells. Now, looking at this very closely, it looks like there's a lot of layers there. When you look at the nuclei, it looks like there's a layer of nuclei there, there's a layer that's over there, there's a layer that's over there. So it looks stratified. However, each one of these cells, on a close-up view of it, each one of the cells is attached to the basement membrane. They're just different sizes. So this is something that looks stratified, but it's not. So we call this pseudo-stratified. Does that make sense? See, it looks like there's a lot of layers there. But in actuality, they're all attached to the basement membrane. There's a schematic one, you can see that they're all attached to the basement membrane. These usually have cilia at its apical ends. We usually see this in the trachea, is a good example. It's in the bronchioles, which are extra tubes that lead off the trachea. Okay? And they do usually have goblet cells with them also, producing mucus. It's in the trachea. We've been talking about that with cystic fibrosis and stuff. It's going to produce mucus to collect the particles and the bacteria, and the cilia are already there, and it's going to push it so it produces the cough reflex, and you can cough out everything. Okay? Question about pseudostratified epithelium. All right, the last one I want to do is something where they're just big and poofy cells. Look stratified, but they're all about the same size, and they're huge, and they're poofy. This is something called, oh, there's pseudostratified, and there's a goblet cell in there, different sizes and shapes, okay? But we have transitional epithelium. Ever take this is something you're probably thinking about right now, a big pillow. Okay? Take a big pillow, it's nice and poofy. You put your head down in it, and your head actually contorts the pillow in a different shape. Kind of stretches away from your head. You lift up your head, and it goes right back to a poofy pillow. It kind of changes in a transitional shape for temporarily. And then gradually you let it sit there, and over hours it'll go back. This is called transitional epithelium. This is found in the urinary bladder. The only place you'll find it is urinary bladder. And, well, actually in the tube that leads to the urinary bladder called the ureter. So what happens here is that when a urinary bladder fills up with urine, it's going to change the shape of these cells to accommodate all that urine in the urinary bladder. Then when you empty the urinary bladder, the cells turn back into these poofy cells. It changes shape. It kind of gets, conducive towards stretching. And that is transitional epithelium. Okay? Questions on that? All right, I'm just showing you transitional epithelium. Now, let me show you, and I'll give you a break right after this. Let me just show you the slides that I took with a camera with your microscope slides, showing all these over here. And this is where it turns into a virtual microscope. Okay? That's simple squamous epithelium at 100x. And all of you are saying WTF, and I would say the same thing too. Okay? You're not going to see anything over there. So let's get up closer. See, I kind of threw away the idea of you focusing and bringing up closer, much faster. This is at 400x. You get up to 1000x. Now you can see how thin this is. Right? There's the lumen. This is going to be an epithelial cell. Okay? Thin, squamous. Okay? I could put this up there and ask you, without this over here, but I would say it's 1000x. I would say, what is this epithelial tissue? Be specific. And you would say simple squamous epithelium. Okay? I could then say, give me a location or an organ that you would find this in. Lungs, blood vessel, kidney. Okay? I could also say, what's the function of this epithelium? Gas exchange, not absorption.
Simple diffusion.
What's that?
Simple diffusion.
Simple diffusion I'll take. Filtration I would take. Any of that is fine. Only pick one. Don't run if you're not being chased, okay? If you do put, let's say, absorption and simple diffusion, you'd be wrong. You see what I mean? You can't give half credit, okay? This is simple cuboidal. People in front can see this, but let's get up closer. Here it is at 1000. You see simple cuboidal? Right here, there's the lumen. There's the cells. Simple cuboidal. You see it? Okay? Ignore the arrows. Okay? This is simple columnar. You can't see anything. It's only at 40x. So get up closer. Still can't see. Ignore the arrow. Here's the lumen. Even a lumen in between here. Get up closer. Now you're seeing the lining over here. Get up really close, and you can see the goblet cell there. You see the cells here, and you see the nuclei that's being pushed down to one side there. All right? So let me go back. I'll show you. So what I'm showing you here, it's like a virtual microscope. There it is at 40x, bam. There it is at 100, bam. There it is at 400, bam. There it is at 1000. It's a virtual microscope. Okay? Just showing you where the features are. Instead of you trying to hunt down all this stuff and you're saying, where am I looking at and stuff. Okay? Stratified squamous. Pretty classic here, but you wouldn't get that at first. We get up closer, and you can see this is the area we're looking at. Many layers, but their cells are different sizes and shapes. But look at the top one. See, they're flaking off. See how thin that is? So this is stratified squamous. All right? You won't have to tell me if it's keratinized or non-keratinized. I won't ask you that in histology. Okay? Dermatology, yes, but there won't be pictures for that. But in histology, no. All right? That's really close up to it. The trachea. This is pseudostratified epithelium. You can see the lumen. So you know an epithelium is going to line the lumen. You get up closer, there's what you're looking at. Can't see much there. Now you can see, start seeing things. That's pseudostratified. It looks like many layers. There's the nuclei. They kind of look like the many layers. But you can also see the wonderful, what is that up there? Cilia. Did you take this course before? All right. You see what I'm saying? It's not bad. Okay? This is transitional epithelium. It's a cross-section right through the ureter, the tube that leads to the urinary bladder. You get up closer, this is all it over here. It looks stratified, I realize that, but all the cells are the same size. This is big and poofy. That's transitional epithelium. Okay? The only thing, before I let you take a break, the only thing I want to just say, there's a few things, but one thing I just want to say right now is if you're going to say, you know, if I show you, let's say, transitional epithelium, you can't say bladder. Because I don't know which one you mean. Are you talking about the gallbladder? Or are you talking about the urinary bladder? It's not that I'm being hard. I just don't know which one you mean. Does that make sense? I mean, that's a big difference. Okay?